Hello, everybody around the world listening to this BioLife conference. I'm Dr. Paul Billings, the Chief Medical Officer of Life Technologies, and I have the privilege of talking to you today about innovative genomic analysis and some of the challenges that confront this. We have about an hour to talk. I hope there'll be lots of questions. You please submit your questions. I'm going to take the questions pretty much at the end so that I'll try to talk for 40 or 45 minutes and then have 15 or so minutes to take any questions that are available. And I'm delighted once again to have the opportunity to speak to you all. Let's see about advancing this. Hold on a second. There we go. So the first slide is a kind of an obligate slide for me since I work for a public corporation. If there's anything that sounds like investment advice or forward-looking statements in this, do not take it as anything but my own personal view of the world, and certainly do not take it as specific investment advice. I am going to be talking to you about our instrumentation, some of the instrumentation for which my company, Life Technologies, produces. We produce, except for a couple of things which I'll identify, research-use-only products. And research-use-only products are supposed to be used in research, not in the clinical laboratory. If I do talk to you about clinical services, for the most part, these services are governed by the rules of CLIA, the Clinical Laboratory Information Act, Improvement Act, and are not FDA approved devices or services. So let me just briefly tell you a little bit about Life Technologies. We're an over 20-year-old company. We have a very enviable position worldwide as, I think, the premier enabler of life science research. We are cited in over 600,000 scientific publications. We have been granted over 4,000 patents, and we have employees based worldwide and have generated revenue in excess of $3.7 billion in the previous fiscal year. So we're a big company. We are in the process of being merged with Thermo Fisher. If and when that closes, we will become an even bigger company with an even larger worldwide reach. Amongst the many, many things that we produce, the many consumables and platforms that Life Technologies produce, and we make about 50,000 items which we sell in our both printed and online catalogs. We make a range of genetic and genomic analysis platforms. And these can be used for the whole range of important analytic targets, from the discovery tools, which would be, for the most part now, our ion torrent line, both the Proton and the PGM platforms, to our confirmatory platforms, and we have two FDA-approved platforms, our Sanger capillary electrophoresis equipment and our qPCR QuantStudio platform. And then, of course, we make a variety of other consumables, library prep, informatics, other things that can be used to support genomic analysis across the spectrum of its activity. So now let's turn, with that background and my coming from a company with a many-year history of interest in both genetic and now complex genomic analysis, let's talk a little bit about what genomic analysis is and what can be done and where the challenges lie. Genomic analysis is not just measuring directly the genes or the gene sequence. It is a conversion of lots of information into something that might be usable in the clinical consulting suite. So family history information is obviously important. Some of the conventional chemistries and biochemical analyses, which were often the traditional way in which genetic deficits were, or genetic anomalies were identified, are still important. Cytogenetics, whether one does it with more conventional dyes or with molecular probes, remain particularly important in identifying movements of large units of chromatin and genetic material. Obviously, the modification of DNA by epigenomic activity is increasingly a source of analytic interest as one tries to identify the impacts of the genome. And then conventional genotyping, looking at directly for mutations, either directly or through PCR-based activities, or gene sequencing, which really is the sine qua non of genomic analysis, because it produces the sequence of the genomic region that one might be interested in analyzing. All of these types of analysis, some with more history, obviously, we've been thinking about family histories for over 100 years, since the time of Darwin and Mendel, really. But we've identified families for millennia that had illnesses in it. But all these other forms of analysis remain an important part of complete genomic analysis, particularly as it enters the clinical setting. Now, we're going to focus, for the most part, recognizing this complexity, on sequencing analysis. And of course, we've seen an enormous explosion over the last 40 years or so in our ability to sequence genes, whether they be in animal or bacterial models, or plants for that matter, or in humans, and to reliably reconstruct the sequence of genes. And most people talk about genetic sequencing as in generations. The first generation was driven by Fred Sanger and Alan Maxim and his colleagues at Harvard. And the Sanger sequencing remains the gold standard of sequencing. Maxim and Gilbert sequencing is still done in many places as well. And of course, Fred Sanger and Orla Gilbert have received their Nobel Prizes in part for their work on sequencing, which was so important in setting the stage for the Human Genome Project and for many of the important discoveries that have been made over the last few decades. Then, around the time of the generation of the Human Genome Project, it became clear that we needed to enhance our throughput of sequencing in order to, and we didn't even really know how big the human genome was at the time, but as we now know, the haploid genome is about 3 billion bases. In order to do 3 billion bases on a Sanger platform would have taken many decades. And so new techniques, often called next generation techniques, were developed. There were several. There remain several that are in commercial and clinical application now. But these allowed for rapid sequencing of small segments and using informatic tools, the reconstruction, accurate reconstruction of large swaths of sequences, and next generation sequencing really drove the completion of the Human Genome Project during the second half of the 90s. We now have, at this time in 2013, a whole series of newer technologies, might be called next generation sequencing or third generation sequencing, and they involve both light technologies that involve light, and all the second generation or next generation sequencing are mostly light driven technologies, or those that use electronic currents or other or other methodologies to detect the incorporation of DNA bases by polymerases. These electronic methods, of which I'm familiar primarily with one, the ion torrent methodology, has some very significant advantages. Those advantages, I hope, will become clear in the further slides. But the point is that I wanted to make by this slide is to say that there has been already 3, and depending on how you divide it, there may be 4 generations of sequencing technology available, and that this innovation has been astounding in its ability to increase the output of a sequence available for analysis by researchers and clinicians. This slide gives you a sense of the pace of this innovation. So let's just say that beginning around the 1960s, there has been an incredible increase in the output of human genome sequence and sequencing in general, and that this output it seems absolutely unebbed. I do not think we have seen the top of the curve, so both in terms of speed to completed sequence and amount of completed sequence in that period of time, we've seen really unimaginable improvement in sequencing. One other sort of measure of this is in one of the electronic forms of sequencing, as the ion torrent method, which simply measures the change in pH as polymerase moves along a genomic sequence and incorporates the appropriate bases. It uses natural bases. There's no modification of the bases. It simply allows these reactions to go on in micro tubes, micro plate kind of format that these chips represent. But the reason that I included this slide is just to show you that over the course of roughly 2 years, every 6 months there has been a log increase in the output of these chips, and we're now at outputs that allow for whole genome sequencing either on a single chip or even multiple genomes, depending on the coverage, the amount of repetitions that you want at any particular site on a particular chip. And it certainly allows for the exome, the express genome, to be easily sequenced on one of these chips, and the time for that sequencing, for the sequencing part, irrespective of the analytic part or the front end where one has to prepare the samples, but the actual time on the machine is measured in hours. When back in the days of where Sanger sequencing was the dominant form of sequencing, you would talk about sequencing times in days and weeks. Now we're talking about minutes and hours for very large outputs. So it's really quite remarkable, this really rapid evolution in power, quality, and output of sequencing. Now, what this, of course, has allowed, this remarkable innovative burst in the genomic sequencing arena, is a very profound drop in the cost for generating a genome or a set of bases of very high quality sequence. And so as you can see, this sequencing activity has exceeded in terms of cost and productivity the output of the semiconductor industry that was dominated by Moore's law. And so genomic sequencing is progressing much faster than Moore's law. Of course, our challenge is not only to drop the cost of generating human genome sequencing, but also to increasingly demonstrate that this increasingly inexpensive activity has value, and that, of course, the value of the information not just the low cost of the data. The value of the information, in my view, is increasingly important. And so that tension between lowering costs, which makes it hopefully broadly available, and increasing value of the information, which might dictate its overall value in the clinical setting and be important in reimbursement discussions. This is an interesting tension which is being played out in the genomic analysis world. But I think the message has to be clear. This is an enormously productive segment of human biology, which is where innovation is producing remarkable results. And not surprisingly, if one looks at next generation and third generation placements of machines around the world, we are seeing increasingly that these machines, as they become more and more affordable, as they become move from requiring large rooms with ventilation and many technicians to desktop models which are almost autonomous in their operation, that we have the opportunity to deliver sequencing worldwide, and that this increasing productivity is reflected by increasing placement, increasing interest, and increasing use. So now let me turn to an example, really. I've just made the argument that human genome sequencing has gone through in the last couple of decades a remarkable technological transformation, which makes the generation of the data easy or easier, but why bother? And there are many examples of why bother. I'm just going to give you a couple. The first is a study that I was pleased to participate in with my colleagues at TGen, the Translational Genomics Institute in Phoenix, Arizona, and also colleagues at US Oncology, a large network of oncologists that practices here in the United States. And we began to look at a series of women who had a so-called triple-negative breast cancer. Breast cancer is a lovely story in the sense that many women who have breast cancer have undergone molecular testing, and they either have a HER2 amplification or have susceptible alleles at their progesterone receptor or estrogen receptor sites, and so are able to have specific targeted therapy directed at their cancers, which has improved the outcome of people with ER positive, PR positive, or HER2 positive breast cancer enormously. And the FDA has approved both these tests and the drugs associated with treating these. But there are a lot of women, including many women in the African American community, who have a so-called triple-negative breast cancer, do not have any of those three markers. And in general, their cancers are more aggressive, and the outcomes of women with triple-negative breast cancer are poor. So we put together a series of 14 cases of women with triple-negative breast cancer, and I'm going to just show you what the power of some of this kind of analysis can be. These women, we had tissue from their tumors as well as normal breast and blood. We sequenced their genomes, their transcriptomes, all their transcribed genes. Findings were confirmed by orthogonal testing by an outside testing agency. And I'm just going to show you an example of one of the cases and what might occur. So this was a 60-year-old European woman who had been given chemotherapy but had a recurrence. This is a picture, a kind of a diagrammatic representation of her genome sequenced. If you look carefully at her somatic tissue, at her tumor tissue, and compared it with her normal genome around in her blood and in her surrounding tissue, we could identify several genes which were over and underexpressed in her genome and appeared to have susceptible mutations and that were of interest to the experts who were looking at this genomic sequence. Looking more carefully at the pathways that were involved, we could see that her mutations potentially were involved in a couple of fairly well described pathways, and the physicians who were caring for her and who were part of this clinical study were aware that some of these pathways were involved in cell survival, other of these pathways are involved in cell proliferation, that mutations in this area could alter the cell cycle characteristics in a tumorigenic or oncogenic way. And they also were aware that there were drugs that were in early development which specifically targeted these pathways. So we searched available databases and found a phase 1 clinical trial that appeared to be interested in targeting the pathways that might be mutated in this triple-negative breast cancer patient. We enrolled this patient in those phase 1 clinical trials, and this is just an example that after a couple of cycles of targeted therapy directed at the mutations that had been discovered in her triple-negative breast cancer analysis, that she'd had a rather profound, significant reduction in her tumor load after her reoccurrence. And so that's just an example of one way in which next-gen sequencing can impact the lives of individuals, and of course can target people for clinical trials, and hopefully those clinical trials can help them do well. A second area where next-gen sequencing is already having a very profound effect is in genetic disease. We're seeing a lot of activity across the spectrum from prenatal settings and even preconceptual settings through adulthood. There have been many technologies which have been applied individually in these areas. All of them are now converging on next-gen sequencing. One particularly important area is in rare and idiopathic, apparently single gene disorders, whether they're in, primarily in pediatric settings or in adults. And here are a couple of examples of cases in which individuals or siblings have been affected by these complex but single gene disorders, and sequencing has allowed for the rapid identification of those genes, and it allows for us to accumulate more knowledge about how these genes impact the human phenotype, but also may suggest important care pathways. In addition, and we're seeing, again, worldwide application of this use of next-gen sequencing, because of the sensitivity and iterative nature of next-gen sequencers, if you target sequences on specific chromosomes, it allows for very sensitive counting of those chromosomes. And if those chromosome material happen to be in the plasma or blood of a pregnant woman, some of those will represent the chromosome material of the fetus that woman may be carrying, and so it can be used in prenatal diagnostic settings. And so we are seeing a proliferation, maybe even explosion, of non-invasive prenatal diagnostic testing using sequencing platforms to count chromosomes, primarily sex chromosomes and chromosome 21, which is associated with the trisomy 21 syndrome. But as you can see from this set of data, it also can be used to do chromosome 18 and 13, which are also sometimes involved in abnormal fetus conceptions. So both in the prenatal setting, in pediatric and adult genetic diagnosis, and in cancer settings, we're seeing an important application of these genomic analysis methods. Now, what are the challenges? Well, first and foremost, there are a number of issues that come just after you run your genetic material through the next-gen sequencer. You need to realign this information. You need to call out whether the information is accurate and you've detected a variant from a normal, or you do not have a variant from normal. And then you have to decide whether that variant has any clinical information or not. So there are many levels of the bioinformatics pipeline which need to be carefully analyzed, carefully reviewed, and carefully controlled. This is an enormous challenge. We, in many ways, and I'll emphasize these in a minute, have not reached those answers yet. And there are many others. I've listed them here in a set of bullets. I'm not going to read each one of them for you, but let me just say that whether you're considering the economics of developing new technologies or validating them or getting regulatory approval, whether how you deliver that information, solving some of these informatic problems, storing the huge amount of data, whether it be in raw sequence, in variant files, or simply in the medical record, provides a series of challenges, and of course setting standards of what is quality sequencing, what kinds of confirmations are required in order to act on sequencing data, since if you're doing, for instance, whole genome, you have 3 billion targets. How many errors are you going to allow if you sequence all 3 billion bases in a clinical setting, for instance? That's an unknown. And so there are a variety of challenges which need to be addressed as this technology makes its way into more clinical settings and into settings where desperate and sometimes very ill patients are using that knowledge. So, let me expand on just a couple of these areas where we need more information or where we face challenges. One is, of course, we need more data about normal and abnormal genetic variation in the population. And in some cases, when we're talking about rare Mendelian disorders, we don't need a whole lot, because if we just have a few people with the same disease and they have this mutation in one or two genes, we're pretty safe in knowing that that's where the money is. But when we get to other less rare disorders, and certainly as we get to common disorders, we need broad-based public participation to know what kinds of alleles are associated with disease and what kinds of alleles are associated with normal variation. Some of these alleles will vary, having to do with their origin and the regions in which we're doing this work. And so we really need global participation in clinical genomic research. In general, participation rates have been small. Happily, we have some repositories that have been developed over many, many years which can help with issues like this, but the problem still remains that the participation rates are low, and some of these repositories haven't been properly consented, so they can't be used for further population data. So we have some challenges around the generation of standard population data and other kinds of population norms. A second area of challenge is around the question of just storing this data. This slide is included just to give one a sense of the size of an amount of storage that one would have to create to really start to do global a high participation rate whole genome sequencing on individuals. Now, happily, we don't have to do whole genome on everyone right away. It's an interesting, important for standardization, important for research, of course, but we can start with more targeted panels with the exome and work out from there. And so we have some time to deal with the big impact of this problem, and I'm encouraged that innovation in storage and in dropping in the cost of storing this electronic information is occurring. But it is a challenge, and it will be an impediment to the proper analysis of large genomic data sets if we don't see innovation in data storage and then a reduction in the cost of data storage. Another area of challenge is, as I've stated before, the need for standards. There are a whole bunch of areas in which we need better standards. Simply, what is a high-quality genomic sequence? What defines it? What controls need to be available? One defines for genomic analysis clinical utility, and what are the data requirements for showing clinical utility? How do you properly compare and competing technologies, or even synergistic or contingent technologies when you are a healthcare system or a payer? And then all sorts of other areas where standardization might be needed, whether it be in research consents, in trial design, or in many of the other aspects of health economic data gathering, which often goes into the decision making of healthcare systems or healthcare payers, or countries for that matter, whether they're going to adopt a test or a set of data. So all these areas are complex areas where we need better standardization. In the absence of standardization, we're going to be wasting lots of time and money, which could be better spent on direct patient care and research. There are obviously, in healthcare in general, and in genomic medicine and in the proliferation of genomic analysis, very significant economic challenges as well. Our sense of right and of equity would say that we should deliver the benefits of genomic analysis at a very low cost, which would allow it to be a universal solution in healthcare. But we also recognize that R&D is a costly activity. We're seeing that as our research budgets are decreasing in the United States and in other developed part of the world, and that we need a return on investment in order to pursue some of these commercial opportunities. We've already talked about the importance of demonstrating clinical utility, and that's an important integral part of the economic challenge. To distribute and support properly this complex information and to provide the electronic databases and records required is time-consuming and expensive. In general, the healthcare environment, the rising cost of healthcare puts a sort of a cold, it's like a cold shower for those of us who are trying to innovate and bring new kinds of information into the healthcare setting, primarily because innovation is viewed as expensive. Even if that innovation is targeted at improving efficiency of the healthcare system and thus eliminating waste or actual cost cutting, innovation is viewed often skeptically. And then in the United States in particular, uncertainty about how one submits a bill and what might be paid. If one does submit a bill properly, this all becomes an important part of the economic challenge. So let me conclude now and then take some questions. I hope there will be some questions. But let me just say that innovation in genomic analysis is progressing and its translation and its application is breathtaking in its speed and its power, and it's progressing rapidly. While the genes and the genome are not the answer to every disease and disorder, it is important to understand the genomic contribution to all forms of illness. In some cases, it may have a significant contribution. In other cases, it may only have a supportive or minor contribution. But to have it be allowed it to be analyzed, to allow the analysis of the interaction of genomic elements within the genome, so-called modifier genes, which may increase or decrease the pathological nature of a pathological variant. These are all enormous progress in our system, and I think we should be very grateful that such progress has been enabled by the innovations of the technologists who have worked so diligently to give us the human genome sequence and all the developments thereafter. In cancer, in idiopathic diseases or unknown diseases, and certainly in prenatal diagnostics, genomic analysis is already being used in the clinical setting. Thousands of individuals have benefited from next-generation genomic analysis, and this is only going to increase. I do think that standardization and regulatory clarity will help foster this translation, and obviously reimbursement is a key element to delivering a return on investment for those entities that are trying to drive this genomic information into useful clinical information in the clinical setting. There are many challenges to this otherwise really dynamic and important story. We want to solve those challenges because remarkable innovation needs to continue. We have many, many unmet needs in the healthcare system. We have many patients who are suffering unnecessarily, who could benefit from genomic analysis and targeted therapies. We have many disorders where we don't even have the beginnings of a therapeutic approach. And we have many people who are misdiagnosed. Now, all these things can be improved in that situation. Revealing data becomes available, is used properly, and benefits all, and is properly valued. So it's very important that we make our data available broadly, that people understand that information, while the cost of developing the data may be decreasing, that the information to make that data highly valuable and into useful information can be extremely expensive and time-consuming, and that we need to put proper value on that. And that the full application of next generation or whole genome sequencing will become universally available in the future if current trends continue and the challenges that I have outlined and others are addressed, and that of course would be a wonderful legacy of the Human Genome Project and all those individuals who have spent a greater part of their careers working on the challenges of translating genomic analysis into common practice in human health and medical care. And with that, I'll say thank you. I see my light flashing, so I believe there are some questions. We have about, looks to me like we have about 20 minutes in order to discuss any questions that might be available, and I will now see what I can do about finding those questions. All right, and there are a couple. Let's see if I can get to them. So the first question that I have, let me see if I can, so there's this one. What, in your opinion, is the primary limitation behind low participation in genomic sequencing? Is it the cost or lack of full understanding of the utility lack of obvious profit potential, ethics, etc.? Well, that's a wonderful question, and it probably depends, and that was offered by a colleague at the University of Alberta, and I very much appreciate that question. In my opinion, it varies a little from setting to setting. First of all, I don't think that we have ever had a public campaign, certainly not in the United States, and I would doubt in many other countries, except for the high participation rates that you see in some smaller countries like Iceland and some of the Scandinavian countries. But we need a public campaign that says that research participation is a public good, that it is beneficial to your family, to your neighbors, and to your community to participate in research. I mean, there's some parts of genomic analysis that simply cannot occur unless we get broad-based public participation in research. And, rather than have it be a legislative or public policy process that gets, enforced by law, and we have, a carrot or a stick that gets, I would like to see an attempt made to educate broadly amongst the public about the value of research participation. I think, in addition, that fear of violations of privacy or of so-called genetic discrimination that might arise from the misuse of otherwise, ethically captured and beneficial research participation. I think the fear of this and maybe some actual instances in which coercive practices have occurred make people concerned that participation in research and genetic research is not safe. I think the risks are small, but I've always been in favor of enhancing the legal protections against misuse of genetic information. I think your genetic information should not preclude you from getting health care or other social benefits, should not preclude you from getting jobs or promotions that you're otherwise qualified for. And I would like to see us continue to be vigilant that knowledge of our genome and of what's the contents of our genome does not get substituted for knowledge of the content of our character and our ability, which are, of course, the major determinants of our success in our lives. And I have worked over my career to try to make sure that people understand the differences between those. I think those are the two major reasons why we haven't seen better research participation. So education and fear, if you like, and both of those are addressable, I think, and should be on the docket of our politicians and our leaders, as well as our scientific community as well. We have another question, and that question is, while much was discussed about economic limitations, any examples of positive economics impacts of genomics that have been clearly demonstrated? After all, the type of information would be crucial to increase chances of funding, increased participation in further genomic sequencing and studies. So what we're asking for is specific examples where the development of genomic sequencing information has in and of itself allowed for clear economic impacts. And the answer is there are several, frankly, and I'll just touch on a couple. The analysis of breast cancer, either by the provision of the HER2 test or in some cases in women with low-grade breast cancers with the provision of tests which allow one to avoid chemotherapy by looking at the expression of a set of genes in those breast cancers and deciding that breast cancer would not benefit from chemotherapy. There have been clear economic studies in those settings, and it is very clear that they have a very significant, the elimination of unnecessary chemotherapy in early stage breast cancer, the elimination of unnecessary chemotherapy in early stage prostate cancer are targets where molecular analysis has already clearly demonstrated a positive economic benefit. In addition, in those situations, and they may represent 20 or 30% only at this point of the applicable cases, but in those unknown disorders, primarily of childhood, sometimes they're syndromic, sometimes they're more focused, where either whole exome or whole genome analysis has provided a clear target, clear diagnostic target and answer for those individuals. That focusing of activity, that focusing of the diagnosis, the so-called resolution of the medical odyssey that some of those patients are on, foretells significant economic saving. Because in the absence of a concrete diagnosis, many of those individuals continue to have utilize lots of health care, lots of imaging, lots of specialized testing, which once they have a clear diagnosis, they do not require. And so in the area of the medical odyssey, the unknown or idiopathic genetic disorder, we have good examples of the benefit of, on a health economic terms, of genomic analysis. I think those are the two primary ones. Obviously, as non-invasive prenatal testing using next generation sequencing comes on board, while it's not a whole genome type of analysis. It is using sequencing technology to change from an invasive test, an amniocentesis test, to a variety of other tests, whether they be fetal ultrasound and biochemical analysis of the fetus contribution in the mother's blood, to a next generation sequencing. I believe we're going to see overall positive economic impact of that technology as well, and all those are important health economic benefits. So there are several that I can point to that are already available, but I think we need better testing and better health economic studies. I actually am in the process of writing a paper right now in which we looked at the clinical utility of a lung cancer risk stratification, early stage lung cancer risk stratification test, and we've showed very significant improvements in clinical utility, but we need better outputs, better ways of capturing the cost of improving that that clinical utility. What are the cost implications? It's easy to assess the cost of the test. It's also not so hard to assess the cost of the chemotherapy, for instance. But what are the costs and benefits and efficiencies around faster diagnosis, better prognosis? We've never had a system that could do that before, and we need better economic signals, better economic data to do that. So I hope that answers that question. There is another one from a colleague, I believe in Saudi Arabia. What is practicably the application of innovative analysis to routine analysis for end users? And also it needs a large community to be trained. And second issue is the cost and economics. Let me just see if I can understand this question. What is the practicability of application of innovative analysis to routine analysis for end users. Well, so I think what you're asking is, why should genomic analysis replace what we have now, the routine analysis, and is, for instance, the training of physicians in the community a barrier to this, and then is economics a barrier to this too? Well, I think the only reason we should replace an old technology with a newer one is because if the newer one provides more accurate information, or if it's only as accurate as the old way, if it provides it in a new format or in a new or faster, allowing for efficiencies or reduction of waste in the system. Otherwise, I'm perfectly happy to stay with the ways that I was trained and the tests and information that I was used when I was a younger doctor in training. But the point is that genomic analysis in many ways is more sensitive, in many ways is more definitive, and allows for more sensitive and specific information about the genetic component in a particular healthcare setting. And so while in some cases that is not currently useful, and those should then thus require more research to decide whether they should be applied, genomic analysis should be applied. But in many cases, particularly as I said, in cancers, in idiopathic or unknown disorders in kids and adults, and in prenatal diagnostic settings, I think genomic analysis is going to become very much the standard and replace older methods. Will it require new training and education for healthcare providers? Absolutely. This data is in many ways foreign and esoteric to even recently trained providers, and so it will require very skillful communication to make those tests and their results useful in the clinical setting. It has fostered the whole area of genomic physicians and genomic healthcare providers, of which I am one, but there are also new types of pathologists who interpret the information as it comes off the machine. So we're seeing new specialization in the medical community to support primary physicians. And in addition, as I said, while the cost of the data coming off the machines is dropping, the value of the information and all the other things that need to require to cause the system to use this information in the most appropriate way is still to be determined, but is certainly more expensive than simply the generation of the data off the machines. And so we're trying to reach a practical equilibrium, which will depend a little bit on the setting in which the information is delivered, whether it be the developed world or the developing world, whether it be in an academic medical center or in a community medical center or somebody's consulting suite. And so all these things will impact what the cost of this is to the end user and how it'll be used. I think that is the end of the questions that I see asked. Wait, no, I think there may be others. Let's see. There is. I have a comment that says, "Thank you for wonderful answers." Thank you yourself. That's my colleague in Alberta. Thank you very much. There's another question. Let me see what that one is. If there is a mistake, this again comes from my Saudi Arabian colleague. If there is a mistake in genomic analysis, how do you counter-check that? Whether we can repeat other analysis easily. Okay, so that's a very good question. So how do we confirm? We're either whether we do a large panel, whether we get a variant that we've not seen before, or we do an exome or a whole genome analysis. How do we confirm that a finding is not a false positive finding? and something that we should take action on. Well, there are many levels of confirmation. First, next-generation sequencing methods allow for multiple measurements of the loci of interest. In other words, when you do a germline analysis of a piece of DNA or analysis of a somatic tissue like a cancer, you're measuring these bases multiple times. And so obviously if you get some variability in those multiple measurements, that would clue you to the fact that there might be an error. So the iterative nature of next-generation sequencing is in itself a control. The second thing I would say is that may very well be true, but certain regions of the genome are more difficult to sequence than others for biochemical and physical reasons, irrespective of the method of sequencing that you're trying to apply to it. And different methods, whether you use the light methods or you're using electronic methods or other kinds of methods to do this analysis, have different biases and are more prone to have difficulty sequencing certain kinds of sequence formations within the genome. So knowing that, you can use some informatic tools to correct and improve and reduce the number of mistakes. But with all that being present, you're still, at the end of the day, going to be delivered a set of sequence, and you may identify variants within that sequence, and you'll be unsure whether that variant is, in fact, a sequencing error, or it's a true variant, and if it's a true variant, is it an important one or a trivial one, having no impact? And yeah, you will not know that. But one of the other things, of course, you can do is apply what we often call an orthogonal method, another technology, another methodology to the same sample, but focus it on that region that you found the variant and ask the question, is the variant present using another method? And of course, that other method has different errors, different, different strengths and different weaknesses, and may allow you to sort out whether you have a sequencing error or, in fact, you have a real variant. It won't help you know whether that variant is important or not clinically, but it will certainly help you decide whether you have a sequencing error or not. So the application of an orthogonal method, whether it be Sanger sequencing or genotyping, or even the application of another next-generation sequencing method following the initial next-generation sequencing method. These are all have been suggested as ways to confirm sequence variation. And I would say that it's extremely important, particularly if you're talking about a high-risk patient or a patient where you're about to engage in important clinical activity, that you do rule out the fact that there are potentially sequencing problems. All right, and then I have two other comments from my colleagues that say thank you for this, and I appreciate their comments. And with that, I think we are at the end. I'll return the podium to our conference conveners with 2 minutes to spare. And I thank you all very much.